Well, students, um, we're going to talk about nucleotides. It would be a small introduction. From there, we'll build upon. So, what are the topics we are going to talk about? We are going to talk about nucleotide bases, purines and pyrimidines. Their types, purines and pyrimidines. Their structure, right? And then we're going to talk about the importance. What other compounds can be formed, right? And then we are going to talk about ribonucleic acid, RNA chain and DNA chain, and about its structure again, and then what is the central dogma for genetic transcription, for genetic code transcription and the formation of transcription, translation, and the formation of another human being or another cell, that is the transmission of genetic code, central dogma. So first we want to start with purines and pyrimidines. The nucleotide bases, they are divided into two forms. Nucleotide bases. There are two types, purines and pyrimidines. The nucleotide bases are of two types, purines and pyrimidines. Now, how do purines look like? Well, they've got two rings. One A ring, that is six-sided ring. This is attached to a five-sided ring. This is the shape of a purine ring. This is the shape of a pyrimidine ring. These are the double bonds. Again, over here you've got there you've got nitrogen over here, nitrogen, N. This is the purine ring, this is the pyrimidine ring, right? Purines, they are adenine, guanine. Pyrimidines, cytosine, uracil, thymine. These are the bases. Nitrogenous bases. Plus ribose. When ribose is added, it forms adenosine. And the other one forms guanosine. Here again you've got ribose added over here. So I repeat the nucleotide bases, the nitrogenous bases. There are two types, purines and pyrimidines. This is the structure of a purine, this is the structure of a pyrimidine. The nitrogenous atoms are present over here. The rest of the atoms of the rings are made up of carbon. Right? So you've got two rings in purines, A and P. And you've got only one ring for pyrimidines. Now, in purine you have got two nitrogens over here and two nitrogens in B ring, while in pyrimidines there is only two nitrogens, and you have got carbon compounds, carbon atoms over there. Adenine plus ribose, adenosine. Guanine plus ribose, guanosine. Okay. Now, cytosine plus ribose, it forms cytidine. Similarly, uracil plus ribose forms uridine. And similarly, thymine, it forms thymidine. Right? Now, addition of phosphate group. So, these are adenine, guanine, adenine, guanine. These are purine bases. And the pyrimidine bases are cytosine, uracil, and thymine. Now, sugar, ribose attached, it forms adenosine and guanosine. And when phosphate group is attached, it forms adenosine. Now it forms adenosine monophosphate. And the second compound is guanosine monophosphate. Similarly, over here you've got cytidine monophosphate. You've got uridine monophosphate. And you've got thymidine monophosphate. So simple nucleotide bases will eventually form RNA chain, and they will eventually form DNA chain. To start with, what are nitrogenous bases? They are of two types, purines and pyrimidines. These contain nitrogen, so they are called as nitrogen bases. Right? Now, this is the basic structure of purine, this is the basic structure of pyrimides. There are minor variations. For example, when you have got adding amino group over here, when there is amino group over here, this is adenine, right? And when there is amino group attached over here, it is guanine. Similarly, you have got cytosine, uracil, and thymine, three bases with minor variations of amino and methyl groups. There are methyl groups attached over here. This is thymine, and if you've got two oxygen over here, this is uracil, and when you've got attached methyl group over here, you form thymine. So these are minor variations which lead to these compounds. You can always check it. So these are nitrogenous bases. This is the parent structure of purines and pyrimidines. Now, adenine, when it attaches to ribose, then it is called a nucleoside. It forms a nucleoside. Ribose is a nucleoside. Nucleoside. This one is a nucleoside. When ribose are attached, nucleosides are formed. Similarly, this one is a nucleoside. the ribose are attached. And when the phosphates are attached, it is called a nucleotide. So these are nitrogenous bases plus ribose, nucleosides, adenosine, guanosine. Over here, cytidine, uridine, thymidine, right? And when a phosphate is attached, they form adenosine monophosphate, guanosine monophosphate. Similarly, over here, you have got cytidine monophosphate, again you have got uridine monophosphate, and then you have got thymidine monophosphate. So this is how you get the formation of important compounds. Adenosine monophosphate, that is AMP. Guanosine monophosphate, that is GMP. Then you have got cytidine monophosphate, that is CMP. Then you have got uridine monophosphate called as UMP, and thymidine monophosphate TMP. These are the building blocks. I'll write again. This is AMP, GMP. And over here you've got cytidine monophosphate, uridine monophosphate, and thymidine monophosphate. So now they are ready to form RNA chain, DNA chain. And remember, there is only one phosphate group attached. If there is plus phosphate, then it will be ADP, GDP. And similarly it will be CDP, UMP, thymidine monophosphate remains as such. And again, if there is another phosphate attached, then you will form GTP. So AMP can convert into ADP, ADP can convert into ATP. Similarly, GMP will convert after addition of one phosphate more, it will become GDP. And similarly, when another phosphate group is attached, it will become GTP. Over here, you've got some compounds, that is, it will form CDP on addition of another phosphate, and it will form UDP. Similarly, on the addition of third phosphate, it will form CTP, and this will be converted into UTP. So this is how the compounds are formed. So this was an introduction. Then what are nitrogenous bases? These bases, there are only 4 types of bases. Adenine, guanine, five bases. Adenine, guanine, cytosine, uracil, and thymine. 2 are purine bases, 3 are pyrimidine bases. Well, over here you can also add certain other purine bases such as hypoxanthine and xanthine, but these are not involved in DNA. but these are also purine bases. But in DNA, adenine, guanine, purines, and cytosine, uracil, thymine, pyrimidines are involved. And first of all, the nitrogen bases convert into nucleosides by the addition of sugar, that is ribose. Then they attach to phosphate, forming mononucleotides over here as well over here. Then when there is second phosphate group added over here and over here, it forms adenosine diphosphate, guanosine diphosphate, and on the addition of third it forms ATP and GTP. ATP and GTP, you know, the high energy compounds. Similarly, you've got cytidine monophosphate, uridine monophosphate, thymidine monophosphate. By the addition of another phosphate group, they form cytidine diphosphate, uridine diphosphate, and similarly cytidine triphosphate and UTP, uridine triphosphate, high energy compounds. These are the building blocks. They have got important functions in the body. We'll discuss next time. And then also I'll tell you how they go on to form RNA and DNA chains. Right? And then we'll discuss the central dogma for transcription. See you next time. Here I go. इतनी सारी थी बाकी